Francesco.
Elegant, very elegant speech, and very also quite clinically relevant speech. So we can move forward on the agenda. It's actually my turn. So I will share the screen. So today, I would like to give you an overview about an emerging class of drugs, which are BET inhibitors. Can you all see the slides? I hope so.
Yeah.
Okay.
Yes,
okay. Thank you. Thank you. So I can't see faces, but some voices help sometimes. So yeah, I would like to shed light on BET inhibitors. Why? Because these drugs are really representing a promising therapeutic tool in cardiovascular patients, and I will try to convince you why this is the case. So I will, of course, repeat something that was already said by Sam, but I think it's always good to repeat some concepts, especially for the people who are approaching the field. Over the course of life, several environmental stimuli and triggers are contributing to the induction of this epigenetic signal. So we have to imagine that epigenetic signals are quite dynamic. Over the course of life, they start accumulating, and this includes changes of DNA methylation, such as CpG methylation of gene promoters and bodies, post-translational modification of histones, and that was actually the topic of Sam's presentation, and also non-coding RNA, and Emma will come to that later on. The good news about epigenetics is that first, these modifications are reversible over the course of life, so they can come, but they can also go upon the cessation of a specific stimuli. The second one is that these changes are amenable to pharmacological intervention, as also elegantly shown by Sam in the previous presentation. The problem with these compounds, which were developed over the last 20 years, is that they are, in most cases, not specific. So they lack specificity, and they are associated with several side effects. Why is that? Just because if you look at how complex is chromatin biology, you can understand that we cannot switch off the transcriptional programs that we want and leave unaltered the other one. So at the end, these compounds, or at least the majority of these compounds, will also affect cellular homeostasis and cell cycle. And this will have also unwanted effect. If we imagine to treat cardiovascular patients, maybe we can also get cancer, just to give a practical example. So if you have in mind the DNA, which is wrapped around histones here, this is a graphical representation of how chromatin looks like. You see here that you have several modifications in terms of post-translational modification of histones. So I just have a pointer, maybe helps to follow the presentation. Here you have this green signal. These are just acetylation sites, methylation site of lysine residues at the histone tails. So these are the tails, and these are the specific lysine that are being methylated or acetylated. They can be also phosphorylated or liquidinated. So in other words, there are families of enzymes which are doing this job of writing specific modifications. So there are writers that are involved in this phenomenon. There are erasers that can remove the methylation or acetylation, histone deacetylases, histone demethylases. And finally, there is a family, emerging family of readers. So what do? At the end, they read. They read, they interpret the biological signals, they eventually bind to this specific post-translational modification, and they start to influence the transcriptional machinery. And I will focus on this family. So it's a pretty huge family, as you see on the left panel here, embracing many different proteins which have the ability to read. What do they read? Several epigenetic signals, starting from CpG methylation, starting from phosphorylation of histone tails, methylation, acetylation. But today, I want to focus on this top right circle here, which includes the bromodomain and extraterminal protein superfamily. And why is that? Because these bromodomain proteins are emerging as important molecular targets in cardiovascular disease. And here is the schematic showing the 4 main bromodomain proteins. So as you see, the overall structure, they are these specific features. So they have 2 bromodomains, so all of them. And these bromodomains are required to bind the specific epigenetic signals of interest. And they have an extraterminal domain on top. So as you see, the BRD4 has a quite long extraterminal domain. This seems to confer an advantage in BRD4 as compared to the other Brahma domains in binding and the efficiency and the affinity of this protein in binding specific acetylation signal. So the question is, what do they bind? As said, they bind acetylated lysine, only the acetylated lysine. So they do not bind methylated, they do not bind phosphorylated. So they are specific for lysine acetylation of histone tails. Every time they see a kind of a package of acetylation motif at the level of histone tails, they can recognize, they can read indeed these acetylated motifs, and they can bind. And this is the pocket of this Brahma domain that was identified by crystallography some years ago. So this is a schematic showing how it works. You see, you have here acetylated histones. The reader protein come around and they read this signal and they bind to this acetylated motif. And once they bind, they start to recruit additional factors, including the transcription elongation factor, which is crucial to influence the activity of polymerase II and to start the transcriptional machinery. So in other words, when BET are activated, they induce transcription of many genes, of course, including many detrimental genes for the cardiovascular system. And we know from in vitro preclinical studies conducted over the last few years that these reader proteins play a central role in cardiovascular disease. They have shown to control different transcriptional programs in different cell types within the cardiovascular system, including endothelial cells, smooth muscle cells, immune cells, cardiomyocytes, and they control a variety of processes, including fibrosis, hypertrophy, sepsis, and atherosclerotic cardiovascular disease. So the question is that what happens when you inhibit such proteins? What could be the consequence? Of course, inhibiting such protein may confer an advantage in the sense that we stop the transcription of specific genes because we impede the recruitment of this transcriptional machinery. And this is what BET inhibitors do. So today, I would like to introduce one of the BET inhibitors, one of the most promising FDA-approved BET inhibitors, which is apabetalol, also known as RBX208, who is FDA-approved since 2006 and is actually being tested in several clinical trials, including cardiovascular trials. So this is why I've decided, of course, I can't focus on all the available BET inhibitors, but I decided to specifically focus on this one. So what do we know about this drug? This drug, it was discovered and synthesized in 2006 by Verdeguer, is a company based in Canada, and was actually initially discovered for its ability to upregulate ApoA, which is a key lipoprotein component of HDL cholesterol. So in other words, this drug was able to raise HDL levels. This was the initial observation. And then from 2006, it was tested in numerous cell and animal models, in model of atherosclerosis and obesity, but only in 2013, it was identified as a potent BET inhibitor. So he has this ability to inhibit all the BET proteins. So then with time, we learn how powerful this drug is in controlling several transcriptional programs, which have definitely something to do with cardiovascular disease and also with relevant comorbidities, which often cluster in cardiovascular patients. So impairment of renal function and vascular dementia. So if you look here, the process, they are very familiar to cardiologists and cardiovascular scientists. So they interfere with vascular inflammation. So we have in mind atherosclerosis. They interfere with cholesterol transport, atherosclerosis as well, vascular calcification, for example, aortic stenosis or vascular stiffness, acute phase response, so CRP, acute coronary syndromes, coagulation cascade. So we have all the cardiology in this slide. So that means that very likely these drugs may interfere significantly with several transcriptional programs which have something to do with cardiovascular disease. But what about cardiovascular events? What do we know so far about the performance of this drug in patients with cardiovascular disease? I want to show you this initial analysis. This is a pooled analysis of phase 2 clinical trial from ASSERT, SUSTAIN, and ASSURE. So in this pool analysis that was published a few years back, you see that apabetalol, as compared with placebo, was able to significantly reduce the MACE, so the measured adverse cardiac events, and with a 44% reduction in the event. So it was quite promising, saying that these drugs in humans seems to work, and the side effects were quite contained, so there was no a measure panel of side effects associated in this study, although with a limited number of patients. And then a big cardiovascular trial came on stage, which is the Beton MACE. And here I'm showing a sub-analysis, a recent sub-analysis of this trial. This trial was including patients with type 2 diabetes with a recent acute coronary syndrome. And in this specific sub-analysis, they split the population, which was roughly including 2,500 patients, in the presence or absence of chronic kidney disease. So you see that the main trial failed actually to achieve the primary endpoint. But if you look at the subgroup of patients with diabetes and chronic kidney disease, you see a quite striking reduction of MACE and also of heart failure hospitalization, really suggesting that this drug could be substantially a powerful tool. And of course, we need much more trials with larger samples. But what can we say? So far, this is something that looks exciting, and although preliminary. The good news here, and I come to the beginning of my presentation, is that the side effects observed in the Beton MACE were not relevant with apabetalol, which was also quite striking for a chromatin drug. We are used to the effect of these drugs, and we know that these drugs in the Beton MACE was only associated with an elevation of liver enzymes, 3.3% in the apabetalol group versus 0.7. And this, of course, was a reason for discontinuation in a small proportion of patients, but still, for a chromatin drug, this is definitely a success. So today, I would like to show you some data from my lab, actually, that we generated on the possible role of this BET inhibitor, apabetalol, in the context of peripheral artery disease. You know, peripheral artery disease is a most important manifestation of atherosclerotic process and often occurs at the level of lower extremities, where it compromises blood flow and leads to an increased risk of limb amputation, morbidity, and mortality. So the burden of the disease is huge, and it affects currently more than 90 million people across the globe, and this is a major cause of disability. This is one of the largest cohort study published in the last few years, showing actually in 2 million people that peripheral artery disease is a major features of type 2 diabetic patients. If you have diabetes, you are very likely to develop this complication. That's why we decided to study this specific population of patients with diabetes and peripheral artery disease. And the problem is that despite advances in revascularization strategies in medication, still the outcome remain poor, and mortality at 10 years can be extremely high, especially in case of symptomatic PAD. Mortality can reach 60%. So we desperately need approaches to boost the new vessel formation and new capillaries, so to increase really capillary density and to secure blood flow to avoid the condition known as critical limb ischemia, which is at the end leads to amputation and increased risk of mortality. So I'm showing some data that we generated in our lab, and data were generated by Shafiq Mohammed, who is a brilliant PhD student in my team, and is also following this webinar. And I would like to show you this data which relates to the ability of BET inhibitors in modulating the angiogenic response in the context of diabetes. So this was a very simple experiment where we were incubating human aortic endothelial cells to normal glucose concentration, high glucose concentration, apabetalol 20 micromolar plus high glucose or vehicle. And this is a live cell imaging showing what happened to the migration of human aortic endothelial cells, which reflects the angiogenic capacity of these cells. You see that in normal glucose, the migration is pretty okay. In hyperglycemia, we are talking about 25 millimole of glucose. This process is completely disrupted, impaired. By contrast, APA with 20 micromolar seems to partially rescue this process. And this is data about the tube formation. So the ability of these cells, you know that endothelial cells form tubes, and this is a main feature to secure the formation of capillary networks. You see that again, that hyperglycemia seems completely to abolish this process, while apabetalol seems to preserve the formation of networks. So this initial data on endothelial phenotyping suggests somehow that BET inhibition may have something to do with transcriptional programs underlying angiogenesis. And that's what we did. So we look at the panel of angiogenesis genes by using a real-time PCR array. So we went for a semi-biased approach, specifically looking to angiogenic genes. And what you see here, when you compare high glucose versus normal glucose, and you look at the transcriptional signatures, we found that hyperglycemia was able to induce an important upregulation of thrombospondin 1, which is a known anti-angiogenic protein, and a massive downregulation of vascular endothelial growth factor, which is a very well-known pro-angiogenic protein. And what happens if I treat cells with apabetalol versus vehicle? It looks like the other way around, is indeed. So you have an upregulation of VEGF and a massive silencing of thrombospondin 1. So then we were actually looking at whether BRD proteins histone acetylation could be involved in our setting. And we started to look at panacetylation of histone 3 in our model, just with the Western blot. And we found that hyperglycemia leads to an increased panacetylation of histone 3 in our model. And this acetylation is not affected as expected by apabetalol, which should not affect the acetylation, but rather the binding of BRD proteins to acetylation motifs. To test the involvement of these reader proteins in our setting, we just knocked down by small interfering RNA, BRD4 in high glucose treated cells, and we use a scrambled siRNA as a control. Interestingly enough, when you knock down BRD4, there is a massive downregulation of thrombospondin 1, suggesting that BRD4 seems to be involved in the transcription of this gene. And by contrast, we found a compensatory upregulation of vascular endothelial growth factor. So that having said, we should know that thrombospondin is a known inhibitor of vascular endothelial growth factor. So if we suppress the thrombospondin 1 expression, then there is a subsequent increase of VEGF level. So then we investigated a little bit more in detail the thrombospondin 1 promoter, so perform a chromatin immunoprecipitation, and we look at BRD4 binding. So our question was, does this reader protein really bind to the acetylated promoter of thrombospondin? Yes. So here you see that in condition of hyperglycemia, there is a recruitment and enrichment of BRD4 on thrombospondin promoter, which is blunted or at least reduced by apabetalol. So apabetalol inhibits BRD4 and prevents the interaction with the acetylation motif. And this was demonstrated by a co-immunoprecipitation here, where you see that in condition of high glucose, there is a massive interaction between BRD4 and acetylated histone. This was the lysin 27, which is one of the most important active chromatin mark. By contrast, when you use APA, you actually prevent this interaction. And as I show you at the beginning of this presentation, when you prevent this interaction, you block the transcriptional cascade. So in other words, you block the transcription of thrombospondin 1. So we were then testing in vivo to see what's the relevance of this in vivo. So we were studying mice with type 1 diabetes, with streptozotocin-induced diabetes, and we perform a model of hindlimb ischemia to mimic diabetic peripheral artery disease. As you see here, we have 4 experimental groups. So we have controls, diabetics, plus, minus apabetalol. So here you see that diabetics show an impairment of blood flow, which seems to be restored by treatment, by oral treatment with apabetalol. And here you see the blood flow recovery in the 4 experimental groups. Again, here you see that diabetic mice show an impairment of blood flow recovery, which is restored by treatment with apabetalol. And this was partially explained by an increase in capillarization within the skeletal muscle of these mice. When investigating the samples, we actually found with this immunofluorescence for CD31 that the number of capillaries and capillary density in general seems to be increased by treatment with apabetalol. When looking at the expression of thrombospondin in these mice, we found also here that in diabetic mice, thrombospondin was massively upregulated as compared to controls. But when you treat them with APA, you can really silence the expression of this angiogenesis inhibitor, which was quite striking and evident. And if you look at the behavior of vascular endothelial growth factor, you see that vascular endothelial growth factor is preserved in control animals, is blunted in diabetics. But when you treat with APA, again, you restore VEGF signaling. And this could be the reason why we observe an increase in capillary density. Again, also in vivo, we tested the binding of BRT4 on thrombospondin promoter that was reduced again by apabetalon. And the co-immunoprecipitation also here confirmed that actually the interaction between BRD4 and acetylated MODIF is attenuated when you use apabetalon as compared to vehicle. Then we wanted to have a look in human samples to see whether these findings may hold true in patients. And for this, we collaborated with Dr. Gaia Spinetti from Multimedica in Milan. And she provided us with samples from patients with diabetes. These are muscular specimens for patients undergoing surgery. And as you see here, when we look at the expression of thrombospondin in diabetics as compared to controls, we found that thrombospondin was significantly upregulated here. By contrast, VEGF was downregulated in patients with diabetes. So we could actually confirm that our pathway was dysregulated in patients with diabetes and peripheral artery disease. And what you see here, we were testing BRD4 binding in thrombospondin promoter. We found a substantial increase in samples from patients with diabetes. So BRD4 recruitment seems to be enhanced. And if you look at the stainings where you see the merge image between BRD4 and histone acetylation, you can see that in patients with diabetes, there is an increased interaction between reader proteins and acetylation MODIF, something that is less pronounced in controls. So in other words, these data show that BET inhibition with apabetalon could be an interesting tool in this patient population. Of course, this is yet to be proven, but it looks like that the epigenetic remodeling at the thrombospondin promoter seems really to call for an action of this drug. So to make a long story short, this is a simple schematic that summarizes these findings. In condition of hyperglycemia, BRD4 binds to acetylated MODIF and enhance thrombospondin 1 transcription. This leads to thrombospondin upregulation and concomitant downregulation of VEGF, thus affecting the angiogenic response. On the other hand, when you block this BRD4, the binding between BRD4 and acetylated MODIF, you block actually thrombospondin transcription, and this leads to thrombospondin 1 downregulation. VEGF is increased, and then you have an increase of angiogenic response. So these findings may open perspective for a personalized approach in cardiovascular patients. A few years ago, we wrote this review together with Sam, Sara Costantino, and other friends. And we were really convinced that if you have a deep look at the individual epigenetic profile in patients, you can really get a lot of information. This is just an example. If you look at specific gene promoter of interest, you study the acetylation pattern, you see that there is interaction with reader proteins. Maybe those genes are good candidates for apabetalon. And of course, if you extend this observation in a more unbiased way, you can really profile the whole spectrum of promoter acetylation and BRD4 recruitment by simply performing a ChIP-seq. So I think that this is a great example of the BET inhibition of precision medicine applied to epigenetic drugs. And we already have some hints related to apabetalon. We could already identify some groups of patients that could really benefit from these drugs. These are patients with type 2 diabetes, with chronic kidney disease, with heart failure, and perhaps peripheral artery disease can be also part of these subgroups of patients who can really have an advantage with this class of drugs. So we definitely are dealing with a possible new kid on the block on top of SGLT2 inhibitors, on top of the GLP-1 receptor agonist. So BET inhibition could really be a next chapter in our fight of cardiovascular disease in patients with diabetes. And I'm pretty sure that the next trials, which already look promising, the interim analysis will show a beneficial effect of this class of drug. Of course, there are many aspects to clarify about selectivity of these drugs and how these drugs will really translate in improved health outcomes. But I think it looks really a promising step toward the future, and epi drugs could be the future. So let me thank my team at the Center for Molecular Cardiology. A special thank to Shafiq, who generated this exciting data for us, and Sara Costantino and Samuele Ambrosini, which were also helping in assembling such an outstanding meeting. and Florian, Era, Elena, so all the people who work with me and the collaborators. We have outstanding collaborators who made this possible, and the colleagues in the clinic, which are important for the also exchange and for the samples from patients. So, and I thank you very much for joining this seminar. Thank you.
Thank you very much, Francesco, for the great talk. Before I go to the questions from the audience, I have a question also to give you. Have you seen, because if I understand correct, the mechanism of action that you think from the expression of this thrombin DHSB gene, if I remember correct, is because the binding of beta protein, and then you see expression, high expression. And when you use the drug that inhibits the binding, then you lose the expression. Have you seen this, if the drug affects the stability of BRD4 or the levels of acetylation? And the follow-up question, what is the effect or interference of CDK9? Because there was a lot of big discussion of BRD4s interacting or not interacting with CDK9, and this has to do with POL2 progression. Have you checked on this? Thank you.
Thank you very much. This is a very important question. We have checked concerning the BRD4 inhibition. We have looked at the interaction with acetylated histones. And so your question was mainly related to the transcriptional control of thrombosponding. So there are some BET inhibitors which specifically degrade bromodomain, and this is not the case of APA, or at least so far, it's not a function of APA-betalon. So we know that there is a growing family of drugs which just are involved in degradation of BET proteins. I think this drug will really be on stage quite soon. APA works mainly by inhibition, which we have to say is a non-selective inhibition of the bromodomain. So I've shown bromodomain 4 because we know that is the bromodomain that with more affinity can bind to acetylated motif, but of course it should have been worth it to invest all the spectra of the other 3 or at least 2, because the other was in germ cells, BRD 1 and 2 in this context. So the other question, we did look at the acetylation that I didn't show for the sake of time. Actually, interestingly enough, we found that APA-betalon reduced histone acetylation, and this is known, it was reported in the past to be a features of this drug, because we know that bromodomain, they also have an acetyltransferase activity. So when you block this bromodomain, you also have a reduction in histone acetylation. So it was an interesting observation that we were expecting somehow, but we actually saw it in cells and in mice. So, and concerning your last question, we haven't checked this. It's a very interesting, intriguing point, because it was really nice to see this. It's highly debated, I would say.
Yeah, it's true. It's true. Thank you, Francesco. Now I go back to the questions from the audience. Seng-Ming Chang, and I apologize if I don't pronounce your name correctly. Follow-up is, have you checked other BET proteins besides BRD4? And I think you already mentioned this, yes, but can you elaborate a little bit more? Thank you.
Yeah, very likely the other BET proteins will be also contributing this, because we know that APA is not at all selective for BRD4. So very likely these BET proteins will contribute. This is, of course, a good question. Now, what we show today, it's a bit too simple to think that BRD4 drives everything, but what we know so far is that BRD4 is the main transcriptional orchestrator among the BRD proteins. But with the sake of completeness, we have to investigate also the landscape of all the BRD. So I agree that this is imperative to really convince the community about the spectrum of this drug and its action on the different proteins.
Thank you. Jonathan Coser, very nice talk. What's the effect of APA on mice glycemia?
On mice glycemia, we measure glycemia. I think we found a slight reduction of glucose levels. We believe that APA definitely has something to do with glucose homeostasis. This is so far was not really studied. There were investigations in many fields, but not in the field of insulin resistance and insulin secretion. So I'm pretty sure that APA, also via controlling the inflammation, may affect glucose homeostasis and insulin sensitivity in liver, in adipose tissue, and skeletal muscle. So I'm pretty sure this could be, if I remember correctly, the effect was not that slight, actually was quite pronounced. And these are data that will come with this paper that hopefully will get published soon. And I would be very happy to discuss this further for in terms of collaboration. I think this is a great topic, also the glucose homeostasis and the BET inhibition. Thank you for this question.
You. Erel Lahal, great talk. Did you dig into the decision of therapy upregulation of the liver enzyme secretion upon APA treatment?
We checked for this, but we could not observe any increase in liver enzymes in our mice. We also have to say that in our model, we treat mice for 5 days in line with previous work that was done also by Verdeguer, where the treatment with this drug was not exceeding 5 or 6 days. Of course, in patients, it's completely different. Of course, we can also be sure that the dose we are using exactly resembles the one used in clinical trials and also the timing. So I'm not surprised to see an elevation of liver enzymes in patients, let's put it this way. And I'm sure that if we prolong treatment in mice, at some point we see some effects. What we know for sure is that 5 days of treatment by gavage with this drug seems to exert a quite potent effect on the angiogenic response. I think the liver toxicity is an issue, especially, so that's why you need big numbers in clinical trial. Otherwise you lose patients and you lose power. This is something that happened in Beton Mace. They published this trial in JAMA 6 months ago, and they actually discussed that this could be an issue that was affecting the power, because if you look at the primary endpoint, we are almost there in terms of significance for the cardiovascular endpoint. Of course, we should avoid discontinuation of the drug, and we should understand why is that.
Okay, then I think we should move on with the talks. Francesco, we'll forward you afterwards the next. I apologize because to the sake of time, we'll need to move on. We will forward you afterwards the questions, and you can contact personally with the people and ask their questions. Thank you very much.
With great pleasure. I would be very happy to get in contact.
Thank you, and apologize for that. Thank you very much. We will move on to our next call. Yeah.
Yes. So let me introduce the next speaker, who is she's also a friend of mine and collaborator. So Maria Cristina Vinci. She's from Italy, from Milan. She's a senior research scientist at Centro Cardiologico Monzino. She's a group leader in the unit of vascular biology and regenerative medicine. And she has a long-standing interest in epigenetics and quite substantial experience in the field, from what I could learn by interacting with Maria Cristina. Her lab works on chromatin modification in hematopoietic stem cells, especially in the context of diabetes and hyperglycemia. I think this is quite an hot topic because the ex vivo epigenetic processing of these cells could really be a next frontier in regenerative medicine. So she will present an update of her research in a talk which is entitled, indeed, Epigenetic Roots in bone marrow-derived CD34 hematopoietic stem cells. So I'm really excited to learn from you, Cristina, about your progress, and the stage is yours.
Okay, can you hear me? Yes, I suppose yes.
Yes.
Okay, thank you very much, Francesco, for your introduction and for your invitation to this important meeting, and also for giving me the opportunity of sharing my research, the research of my group with the very experts in the field. So just a very short introduction concerning the topic. Hematopoietic stem cell, as the name per se explain, generate all the cells of the blood and are characterized by the expression of casein differentiation 34. This is a transmembrane phosphoglycoprotein whose function is still partially unknown. However, we can consider this marker a pan marker because under this marker there are comprised an heterogeneous stem and progenitor cell population characterized by different status of differentiation and also of commitment. However, this marker is also expressed by vascular associated tissues and non-hematopoietic cells, both in physiological and also in pathological conditions, such as in tumors. But what is the origin of these cells? These cells during the embryogenesis arise from hemogenic endothelium, and they migrate in different organs, the bladder and liver, reaching at the end of development the bone marrow where they reside. However, during all the adulthood, these cells maintain a very strong migratory activity, since they continuously move from the bone marrow to the peripheral blood and vice versa, taking part to the so-called stem cell circulating stem cells that comprise different progenitors with different commitment, including also endothelial progenitor cells. But what is the physiological function of this movement of the cells in periphery? Within the bone marrow, they maintain the hematopoiesis, of course, but there are hypotheses about their role in the periphery. Since that they have an important role in the maintenance of the homeostasis of the whole organism by contributing to the immunosurveillance and also by contributing to the regeneration of the peripheral organs and tissues. And this is particularly true for the cardiovascular systems, maybe because of the ontological origins. Thanks to these properties, over the years have been developed three major areas of research. The first is aimed to understand the physiological role of the cells, understand the mechanism, how they can induce angiogenesis and endothelial repair. And the second, dependent on the first, is aimed to optimize the use of these cells in cell therapy. Contemporaneously, the third research field is aimed to understand which are the correlation between the number and the function of these cells with with different pathologies and risk factors for diagnostic and prognostic purposes. To this regard, already almost 20 years ago, Hill demonstrated that there was an inverse correlation between circulating endothelial progenitor cells that now can be considered progeny of hematopoietic stem cells and vascular function and cardiovascular risk, transforming these cells in a surrogate biomarker of cardiovascular health. However, more recently, also has been highlighted by this meta-analysis of Paneni, it's clear that hematopoietic stem cells recognized as CD34 positive and CD133 positive cells are stronger biomarker of adverse cardiovascular outcomes. And this is particularly true for diabetic patients. But why these cells are so affected by diabetes? Because the diabetic microenvironment can affect progenitors, can induce a progenitor's perturbation that unfortunately persists also in the cured diabetic patients. And this phenomenon is known as hyperglycemic memory. What characterizes diabetic patients is the chronic inflammation and immune disturbance that together take the name of diabetic immune paradox, and also micro and macrovascular chronic complication. In all these alterations take part in the dysfunction in hematopoiesis, so hematopoietic abnormalities, and also stem cell drift in their differentiation and in the loss of their vascular trophic effect, so with an impaired regeneration of damaged tissue. Moreover, there are recent papers demonstrating that diabetes can induce dysfunction of the bone marrow that morphologically can translate in the substitution of the hematopoietic tissues with the adipose tissue, with the rarefaction of the vascular tissue, denervation, and also with the depowerization of hematopoietic stem cells. Based on this pathological view of the niche, our hypothesis was that hyperglycemia, that is the hallmark of diabetes, can promote a differentiation drift of these cells toward more harmful progenitor cell population that can pathobiologically contribute to diabetic complications. In the last years, my group focused his research on understanding if epigenetic mechanism could be involved in the reduction of the vascular trophic properties that lead to an impairment of their regenerative function, and also if the same mechanism can be involved in the increased meliopoiesis that is observed in diabetic patients and that can contribute to the inflammatory status of the diabetic patients. We took advantage from an in vitro diabetic model based on the use of a CD34 stem cell isolated from the cord blood, and all our data then were then validated on a human sample. In particular, we use a CD34 stem cell isolated from sternal bone marrow biopsy of diabetic patients underwent to bypass surgery. So in this paper that we recently published, the data seems to support our hypothesis, since we found that bone marrow CD34 cells isolated from the bone marrow of diabetic patients already display a transcriptional signature that can be predictive of their progeny dysfunction. In particular, the gene ontology analysis observed the numerous overrepresented biological processes in diabetic patients that were mainly involved in innate immune system differentiation, host defense, impairment of the chemotaxis, confirming in this way that the diabetic immunoparadox can take place already at the level of the bone marrow. So going forward, we also demonstrate that epigenetic mechanism can contribute to the impairment of 6F4-STF1 axis. This axis is particularly important for the regenerative properties of the cells because it's involved not only in the mobilization but also in the migration and in the homing to the tissue of at the level of a tissue injury. So through our in vitro model, we demonstrate that high glucose affect the proliferation of these cells and also the oxidative state with increase of the RAS production that was associated with the reduction of a protective antioxidant enzyme such as NSOD, CAT, and with the increase of the expression of P66 gene. Not only. Hyperglycemia was also able to reduce, impair the migration of these cells over the stromal derived factor 1. And this impairment was associated with an increased DNA methylation of 6F4 promoter. Interesting, this was not the only epigenetic modification, since we found that this modification was associated also with a more inactive chromatin conformation, since we observed an increase methylation of lysine 27 or histone 3, and instead a reduction of pan-acetylation of histone 4. Importantly, all these data were validated in CD34 stem cell isolated from the bone marrow of diabetic patients. Here it's important to know that these differences were present regardless the presence of coronary artery disease as comorbidity. We found a downregulation of 6F4 receptor, and as observed in our in vitro experiment, there was an increased methylation of DNA promoters. But we obviously think that this is not the only gene involved in the impairment of their regenerative function. Other genes likely are involved. So considering that hematopoietic stem cells are ancestral precursor of endothelial progenitor cells, we differentiate these cells toward the pro-angiogenic phenotype after exposure to a high glucose concentration, and we observed that they were less prone to form colony formation, to incorporate in tube formation in vitro, as well as we observed a reduction in the expression of eNOS gene that is considered a marker of differentiation toward endothelial lineage. Moreover, this reduction was also associated with the decreased release of nitric oxide. Here it's important to note that all these defects persisted despite the return of the cells in normal glucose conditions. We focus on, at this point, on eNOS, on this gene, because from the literature it is known that the nitric oxide play an important role not only in the neovascularization, but also in differentiation of these cells by promoting the mobilization, migration, the homing, the differentiation through paracrine and the autocrine mechanism. So to the same, we analyze the promoter, and we found that this is a particular promoter because it lacks the canonical Tata box, and it also is not located close to CPG island. Instead, it is regulated by the so-called regulatory regions, in particular shear stress responsive element, camp responsive element, and a positive regulatory domain that in non-endothelial cells are really heavily methylated. So this means that this gene is just regulated by DNA methylation. So through a method that we developed in our laboratory and we recently also published, we demonstrate that the endothelial progenitor cell exposed to hyperglycemia display a reduction in the demethylation this time of eNOS promoter. And this effect was even more evident in the cells that were returned in normoglycemic conditions. Obviously, all these data were also validated by Sanger sequencing, that is the gold standard that confirm our data. So at this point, we move to the other part of our study. So to understand if hyperglycemia can epigenetically propels the pro-inflammatory differentiation of these cells. We perform a characterization, a phenotypic characterization of the cells after their exposure, and we saw that they acquire a senescent associated secretory-like phenotype that was characterized by reduction of the cell proliferation, increased expression of cyclin-dependent kinases inhibitors, and also by the increased release of pro-inflammatory cytokines such as tumor necrosis factor and interleukin-6 that are involved in the inflammatory processes of diabetic patients. Interesting, we found also an upregulation of P65 subunit of NF-κB transcription factor. This transcription factor is important in numerous inflammatory processes, including in cardiovascular complication of diabetic patients, and is known also to be epigenetically, to undergo epigenetic modulation, as already Professor El-Osta showed several years ago. Consistently, we found that the chromatin close to the P65 promoter had a more open conformation, since there was a reduction of inhibitory histone mark, trimethylation in lysine 9, and instead, on the other hand, an increase in the activatory histone mark, monomethylation of lysine 4. This data was also confirmed by the increased recruitment, so engagement of the methyltransferase Sept7 enzyme. But epigenetics is not only is able to modulate the expression of this transcription factor. We found that also regulate the activity of the transcription. In fact, we found an upregulation of these acetylases that are also called PCAF that play an important role in the stabilization of the transcription factor at nuclear level by acetylating the lysine 310. This data was also confirmed by the translocation and also from the increase in binding activity of transcription factor at nuclear level. But the question is, after this hyperglycemic priming, what happened to these cells when are induced to differentiate toward the myeloid lineage? We use this protocol in vitro, already established and published, that, and we saw that CD54, we didn't, at the beginning, we didn't observe any differences in the total amount of monocytes after the differentiation, but analyzing the SOC population, we observed a significant increase of intermediate monocytes. These monocytes, identified by the expression of CD14 and CD16, are recognized as more aggressive monocytes and inflammatory monocytes that are involved in the atherosclerotic processes in diabetic patients. Moreover, it's important to note that all the population, once stimulated with LPS, released higher amount of pro-inflammatory cytokines. Obviously, we try to understand also if these effects were present also in the CD54 stem cell or diabetic patients. So we validate our data, and as you can see, analyzing the phenotype of the cells, we observe that CD54 stem cell of diabetics were characterized by a senescence associated secretory phenotype. And what is important is that when we differentiate these cells toward the myeloid lineage, the differences were really striking, because in this case, differently from what we observed in our in vitro model, there was an increased myelopoiesis. Moreover, the differences in the subpopulation was even more evident. And again, the stimulation of these cells promoted a very high amount of pro-inflammatory cytokines. So, concluding, what is the take-home message of our research? First is that bone marrow is evidence that the bone marrow is a target organ for the chronic diabetic complications. That diabetes can change the differentiation program of the progenitor cells that lead to the formation of harmful progenitors with defective vasculotropin function and instead excessive pro-inflammatory phenotype that can contribute to the pathogenesis of diabetic complication. Unfortunately, so far, the achievement of a good glycemic control remain the best primary way to prevent diabetic complications. So we hope that the better understanding of the genetic mechanism and the cellular mechanism involved can pave the way towards new therapy and new treatment to slow down or at least reverse diabetic complications. So I want to thank you, my unit of vascular biology and the regenerative medicine, in particular Professor Giulio Pompilio and Erika Rurali, and also all my group of cardiovascular epigenetics, the girl who performed most of the disease experiment, Annalisa Sforza and Raffaella Rinaldi, our diabetologist, Stefano Genovese, and of course, all my collaborators who contribute to this project, in particular Francesco and Sara Cosentino. So thank you very much for your attention.
Thank you, Maria Cristina. It was really exciting. And I think this is, as I say, this is an hot topic. And by the way, it's always nice to see when you acknowledge your team. It's always something that it's nice to see in science in general. So you have a very nice team, and well, the data look very promising. I have a quick question because I would like to leave the stage to our colleagues. What do you think about the role of glycemic spikes in the maintenance of transcriptional memory? So do you think that it's just hyperglycemia per se, or do you think that glycemic variability may rather contribute to maintain chromatin in a detrimental, let's say, structure arrangement?
Okay. In my opinion, the hyperglycemic variability has a stronger effect on the change of the transcription of the genes, and so also at the epigenetic level, because as you can see from the results, the effect were even stronger when the cells return to the normoglycemic condition. So when they are exposed to chronic hyperglycemia, there is a kind of adapt, so that they become used to stay in a hyperglycemic condition. Instead, there is a stressful environment, so there is an increase of radical oxygen species when they have to balance between hyperglycemia and normoglycemia. So I think that the damage is higher.
Yeah, I agree with you. That's why and that was also along my. So we have a question from Dr. Ronda, which is pretty much along the same line. So could you elaborate on the seemingly reversible effects seen in hyperglycemia patients? Irreversible effects, yeah.
Now, reversible, I mean, these are patients that are already in therapy. So they are all patients with were already in normoglycemic condition. So what we observed was the hyperglycemic memory, and the effect obviously was even stronger because in vitro you can reproduce just only some of the modifications, some of the effect of the hyperglycemia. But in the patients, they are exposed to glycemic variability, hyperglycemia for a very long time. But regarding the patients that we enrolled in our study, were all in normoglycemic. We can say that they in normoglycemic condition.
Great. An additional question from Dr. Sabo. She wants to know whether you have performed bone marrow transplantation in your experiments with or without respective epigenetic treatment strategy to clarify the role of bone marrow in diabetic cardiac and vascular abnormalities.
So to use in mouse model?
Yes, in mice, sorry. Yeah.
In mice. So the problem of this approach is that we are now using biopsy, so sternal bone marrow from sternal biopsy. So the amount of cells is very low, a very few cells. So we are planning to use other kind of samples, but yes, this could be a good idea, I mean.
Yeah, it would define the causality even more. I agree. So the question may be in the chat, and yeah, there is an additional question from Dr. Kadri. What about the heterogeneity of CD34 positive cells? Do you think that there is a change in the composition of CD34 population under high glucose?
Change of the expression. So we didn't observe a significant change of the CD34 expression both during the treatment and also in the patient. We have to say that we use the CD34 stem cell from the patient immediately after the withdrawal. So we perform panel of different markers to see the purity of the CD34 stem cell population. And the other markers such as CD14 and lymphocyte marker were not still present. So we didn't see any. At the moment, we didn't see any differences in the expression of CD34. Also in vitro.
Yeah. So thank you for this exhaustive explanation. If there are no additional questions, I would like to thank you for your contribution to this symposium, for the exciting data, and I would like to move further with the next presenter, who is also a collaborator of mine and we work in the same city, in the same university. I hope Francesco could join. Yeah, he's there. Hi, Francisco. So it's my pleasure to introduce Francesco Verdeguer. Francesco is an assistant professor at the University of Zurich, works in the Department of Molecular Mechanism of Disease, and he studied in Valencia, and then he did a postdoc in Harvard, in Karolinska Institute, and has a long-term interest in chromatin remodeling and metabolism. And Francisco's lab currently works on transcriptional regulation of thermogenesis, adipose tissue function, and also in the cardiometabolic disturbances and the crosstalk between adipose tissue and cardiovascular tissues. So his talk today will be about transcriptional control and chromatin remodeling in energy balance and cardiometabolic diseases. So I'm really curious to see your talk, Francisco, and I would like to give you the word, and thank you for being here.
Thank you very much for this kind introduction. And let me just quickly swap this place. I guess you see now here the full mode presentation, right?
Yes, I do.
Perfect. So good. So yeah, again, thanks a lot for this nice introduction. As you said, we live also in the same city. We see each other quite sometimes, and I'm happy to introduce you my research area, which is basically a basic research approach in understanding transcriptional regulation of energy homeostasis. And as you also advanced already, I'm interested in understanding adipose tissue biology. Particularly, I'm interested in understanding how energy balance is maintained. We know obviously how energy intake and energy expenditure are crucial to maintain the equilibrium to maintain energy homeostasis. Of course, this is a complex question that has to be addressed in many different from many different perspectives. And we are looking this at a specific angle in energy expenditure. And energy expenditure, one of the key components of it is the activation of brown adipose tissue. Brown adipose tissue is an organ that has attracted recently a bit more attraction, but has been well studied before in the context of thermogenesis. So mammals and small rodents in particular, but basically almost all mammals perform thermogenesis to control body temperature in cold conditions. And this is thanks to the high amount of mitochondrial content of these cells, which is, as I said, this particular form of adipose tissue that undergoes thermogenesis. And the interesting thing is that it consumes a large amount of energy, particularly glucose and fatty acids. So how we know a bit more nowadays of how brown adipose tissue is activated. Classically, this was known for many years that obviously cold exposure triggers thermogenesis, and this is through direct innervation of adrenergic inputs into brown adipose tissue, releasing catecholamines, but also hormone activation, T3. This is classical activators of thermogenesis. At the end, one of the key events is the activation and the upregulation of a protein known as uncoupling protein 1, UCP1. And these triggers, and I will show you later, exacerbated catabolism, as I said before, free fatty acids and glucose. Mechanistically, how this works is that UCP1 sits at the inner mitochondrial membrane, and this protein uncouples the oxidative phosphorylation from ATP production. This results in a high nutrient demand and a high catabolic flux that results in due to the inefficient ATP production because of the uncoupling. So at the end, this activity, metabolic activity, is highly increased, and this is what generates heat at the end as a byproduct. We also know pretty well the transcriptional activation pathways. I'd say pretty well, but there are still many unknowns. But classically also it is well understood that pathogenic signaling activates PK signaling, and this leads to different signaling events leading to the activation of a known transcription factor known as PPR gamma, as you see here, that is also co-activated by a well-known co-activator known as PGC1 alpha, highly expressed in brown adipose tissue, and this triggers at the end the activation of UCP1 in brown adipose tissue and activation of thermogenesis in brown adipose tissue. As I said before, this tissue has attracted a lot of interest in the last years because until some years it was only thought to be present in this tissue, and this thermogenesis was thought to be present only in babies and animals. But a few years ago, actually already about 10 years ago now, some groups identified the presence of highly active glucose uptake deposits in some individuals. That was identified a bit by chance in cancer patients, but they found that actually this was not tumor cells, but actually was deposits of brown adipose or thermogenic adipocytes, let's say. And you can see here in after later cold exposed individual how this glucose PET tracing highlights really strongly activation of thermogenesis or at least glucose uptake. And as I said, this was shown later that the deposits coincide with thermogenic adipocytes because they also express UCP1. And this also was correlated highly, that was already shown before, that it correlates with an improved metabolic homeostasis. And also there's a recent paper from the lab of Paul Coyne in New York in the Rockefeller Institute where they did a retrospective analysis of thousands of patients, and they clearly showed that having brown adipose tissue correlates with an improved or type 2 diabetes phenotype, decreased coronary artery disease, congestive heart failure, cerebrovascular disease, or hypertension. As you can see here, also you see that with age, the amount of brown adipose tissue is declining, the amount, or I would say also activity, and you also see that actually amount, or let's say patients or individuals who have positive brown adipocytes are very low. I would say you can see here the percentage of amount of adipose tissue brown adipose tissue is very low, and as I said, dramatically decreases with age. So one of the attractive nowadays therapeutic approaches would be how we can unlock thermogenesis and to prevent potentially metabolic disorders, including type 2 diabetes, insulin resistance, and cardiometabolic disturbances. And this has, as I said, attracted many different labs around the world, and many different approaches are taking over. And in our case, we are trying to understand this as from a basic research standpoint, and trying to understand metabolism, and particularly how nutrients and hormones modulate anabolic and catabolic pathways in the context of transcription factor regulation and activation. For some years, I have also been interested in understanding a particular transcription factor as a model of transcription activation and as a model of metabolic regulation. And this is yin-yang-1, YY1. This is a well-known transcription factor in a way because it activates or controls many different biological functions, immune functions. It's involved in cancer. It has kind of a pleiotropic functions. How these transcription factors play specific roles is a bit less understood, and also how in the context of metabolism is also less understood, although I have contributed with some studies to understand the role of YY1 in different metabolic contexts that I don't have now time to explain. But one of our questions was and still is actually how nutrients or hormones can control or modulate the activation of the transcription factor through, for example, through post-translational modifications, and how this in turn could control different metabolic functions. And as I said, we use as a model adipose tissue in the control of anabolism and catabolism. Adipose tissue is also, in my perspective, an interesting tissue to understand this modulation between anabolism and catabolism, because it has a strong anabolic phenotype, anabolic processes to uptake glucose to make fat, and at the same time also it contains or harbors the capacity to burn, at least by brown adipose tissue, to burn a lot of nutrients and play with strongly with catabolic pathways. So our approach was to identify post-translational modifications in response to hormones or nutrients. And we did the first approach by doing a mass spectrometry approach by stimulating brown adipocytes with forskolin. Forskolin is an activator of adenylylated cyclase, which leads to high levels of sialic AMP. And this is a classical way, as I said before, how thermogenesis can be activated. So we looked at the interaction between sialic AMP signaling and WAB1, and how this could lead to differential post-translational modifications. And interestingly, we found many different post-translational modifications, but we focused particularly on one for some other reasons that I don't have time to explain. But we saw that the serine 120 in the transactivation domain of WAB1 was reduced upon forskolin induction by mass spec. We also confirmed this by a Western blot, and we saw that stimulating brown adipocytes with forskolin leads to a strong or decrease in WAB1 phosphorylation at this residue with specific antibodies. And so the model, the observation at this stands as a sialic AMP induces the dephosphorylation of WAB1 at this residue. This was in vitro. We also were interested, of course, in validating this in vivo, and we showed that mice exposed to cold for several hours induce also a dephosphorylation at this the same residue upon cold exposure. We also looked at the other way around. How could be that other stimuli, such as high-fat diet, which is more anabolic prone, would regulate potentially this serine residue? And we saw that upon high-fat diet, we saw actually the opposite, a strong increase in phosphorylation of WAB1 at serine 120, as you can see here by the model. The question then is, what is the role of this phosphorylation? What does it make downstream effect, and how also it's regulated, the players that control this phosphorylation? The first question, what is the role of this phosphorylation? One of our first obvious question was whether it would be involved in thermogenesis, and actually we showed that by doing point mutation of serine to alanine that it is not possible to be phosphorylated, then we see actually an increase in UCP1 expression. And that was very clear also because when we did the phosphomimetic, there was not such a upregulation of UCP1 at the protein level, but also we saw at the transcriptional level. And this is also correlated with the functional assays that we looked at oxygen consumption as a way to understand metabolic function and thermogenesis. And we see also here that the point mutation of serine 120 to alanine leads to a higher oxygen consumption in brown adipocytes. We wanted to go further, obviously, to understand a bit more in a wider approach, what are the consequences of WAB1 dephosphorylation by RNA-seq, and we saw many changes, and I quickly summarize here the main pathways, because we suspected and we saw that actually many pathways by GCA analysis that were upregulated are actually catabolic pathways. So as you can see here, TCA cycle, fatty acid degradation, glycolysis, and so on. And many pathways that were downregulated by the dephosphorylation were actually anabolic here, mTOR signaling or basal transition factors or ribosome biogenesis. And this is somehow the same, but in another analysis. You also see here thermogenesis is upregulated. I don't have time to go that in depth, but I just quickly only show you how we try to understood the signaling and at the signaling level, catabolism and anabolism, and we looked at classical mediators of these processes such as AMP kinase or mTOR. And as you can see here, AMP kinase is highly activated by serine 120. And in this case also, and that's what we think it happens. So there is a catabolic gene program that is controlling also AMP kinase in a positive way. On the other hand, actually, we also saw mTOR, phospho-mTOR, and phospho-70 kinase, which are also activators of anabolic pathways, which are also here activated. This we can explain, and we think that it happens. What happens is that we see actually a decrease in ribosomal gene expressions, as we also saw by the RNA-seq. We think that this leads to a hyperactivation of mTOR due to the lack of potential activation of ribosomal biogenesis. So this is actually downregulated, and there is a hypercompensation mechanism to activate this translation. But anyways, this is a bit too, I don't have time to go to that depth in detail. But what we think it happens, at least our model hypothesis, is that YY1 can play a switching role between anabolic and catabolic pathways, particularly by modulating this phosphorylation. So when Y1 is dephosphorylated, it triggers catabolic pathways, as you saw UCP1, but protein, but many others as well. Then obviously what we wanted to understand is actually how is this happen? How Y1 controls these pathways at the transcriptional level, as I saw you saw UCP1, but how this is controlled mechanistically, right? And we did ChIP-seq analysis of YY1 versus stimulated YY1 cells. So in this case, we use phosphorylation stimulation to mimic the dephosphorylation. We also have done now the point mutation for the ChIP-seq, but for now I show you just the cells were stimulated with just control DMSO or forskolin, and as we see here the binding of YY1 across whole genome, and you can see here that actually there is no major changes. Not actually seen here, at least in this graph here is plotted all genes centered at the transcription start site minus and plus 5 kb. And you see here that actually most of the bounded sites are in the promoter, around 60%, and they also binds to the classical known motif of YY1. However, interestingly, also we saw that many of these genes that are where YY1 is bound, actually also there is an enrichment of thermogenesis and ribosome, again here thermogenesis, so it fits also with what we saw. However, what we obviously wanted to look at more precisely is obviously at UCP1, and you see here how the profiling binding of YY1 upon forskolin stimulation at the UCP1 locus. We see here that UCP1 is stronger. There's a higher recruitment of YY1 at this enhancer site, and you also actually think that this may be a novel enhancer where YY1 binds here. We see that this other classical enhancer of UCP1 here also this is a higher enrichment at minus 2 kb, but here there was a stronger also enrichment. This also we confirmed by ChIP-qPCR. Here you see also a stronger enrichment, and also just to clarify, we also saw that using the phosphorylation antibody that detects only the phosphorylation of CD120, then we don't see anymore the changes in binding because this only detects of the phosphorylation. So just to have an additional control. In the context particularly luciferase reporter assays, we also saw that UCP1 expression is increased, or reporter assay is increased upon the mutation of or the point mutation of serine 2 alanine mutation. Here the story is not complete because we wanted to understand a bit better, more precisely, what are on one way the direct targets of YY1, and to understand a bit more precisely how YY1 regulates these pathways. So here what we showed and we tried to do is to plot all genes that are downregulated or upregulated by the RNA-seq. So just crossing the data set from RNA-seq and ChIP-seq and showing again here only the genes that are regulated by RNA-seq, plotting the ChIP-seq binding. If you focus now just on the downregulated genes, you see here that this actually shows from all genes that are downregulated, which are direct targets. And you see here that there is no major changes here from control or forskolin. And you see here that this I would say will coincide with the direct targets of YY1, because this is where YY1 is bound, at least at the start site. And again, the message here is that these genes are downregulated in the forskolin condition. However, there is no change in binding. So we think that this is an interesting aspect. And we also saw that these actually genes coincide mostly to ribosomal and translation genes. This is an example, elongation factor 4B, and this is the ChIP-seq binding, or the profile binding. It's just a strong enrichment, and there is no change in binding upon forskolin treatment. And this is again actually the RNA-seq actually, even though there is no change in differential binding, there is a change in transcription. So there's something else that should control the transcription in this case. So this is what we think it happens for this case of this particular set of genes. When there is phosphorylation, these genes are actually activated, and when there is no phosphorylation, these genes are stopped. But again, this is regardless of the Y1 binding, and we think that this may be important for recruitment of different cofactors that then may be controlling the transcriptional activity of these genes. And we also have to remember that actually this phosphorylation site, or this serine 120, is located in the transactivation domain, suggesting that there might be also, again, coactivators that may control this function. But we thought that this kind of was interesting. On the other hand, also we wanted to see what actually, the changes that were the changes in binding that were made by forskolin treatment. And here we selected only, of course, the binding sites that were differently bound. And here it is not centered by the start site. This is just anywhere, any gene that any locus that was changed. And you can see here actually that by forskolin we see all these genes that have all these sites, or these binding sites that have a stronger binding of YY1. Interestingly, most of these sites coincide actually to distal intergenic regions. Around 40% are actually distal intergenic, or other introns, here 25%. So these are not really close to the promoter site. And among those target genes, we found among the top hits actually the two interesting targets, which are ADCY3 and PRK2B, which I will come a bit in a minute. Just to show you here the profiles, we also see here how there is a stronger binding of Y1 upon this forskolin treatment. And again here for ADCY3, how the binding is much stronger. So here, this actually is quite far away from the start site. It's around 20 kb and also 40 kb distance from the start site. We suggest that this again is still anyways controlled the transcription activity of YY1, but this may involve a reorganization of the chromatin and distal or chromatin looping. And this was what we did using actually public databases. And anyways, also we have done the experiment. Now we are analyzing it, is to try to see whether these regions where Y1 binding site is, if it binds at the genomic level to the transcription start site. And we can see by this Hi-C experiments and Hi-C data sets that actually there is a stronger, or somehow suggesting a binding of Y1 to these sites. In this case, for PKR2B and ADCY3 as well. And you can also see here that the transcription activation, actual transcription of the gene is also upregulated in this case. And in this case, ADCY3 is actually highly upregulated by cold exposure. In fact, Y1 has already been involved in the formation of this chromatin looping by a paper actually from several years ago from Rick Young, and they also showed that this is similar to CTCCF. Y1 may be also involved in this looping to control transcription activation by binding to distal regions. So that's one of the ideas that we are following at how mechanistically it could work in some particular targets. So we could define somehow these class A targets in the context of ribosomal genes that I showed you before, where the phosphorylation triggers anabolism and the dephosphorylation triggers the suppression of these genes, but without changes in DNA in chromatin recruitment of YY1. On the other hand, we would define these other class B targets where actually then yes, here the phosphorylation triggers a change in YY1 binding to these sites, and in this case, the phosphorylation triggers a recruitment and potentially also this chromatin looping formation to activation of these key genes that I will explain quickly right now. We also notice a slight change in the DNA consensus motif of YY1 and seems to be a bit more specific when there is this kind of distal regions compared to the classical start site here, or the proximal promoters. Going to these genes, PRK2B and ADCY3, actually they are highly expressed in brown adipose tissue, also other tissues, but you can see here in red, they are highly expressed in adipose tissue and brown. This PRK2B is actually a catalytic activity of PKA, and ADCY3 is a component of the protein G receptor. So it's adenylated cyclic, and it's a component of the activation of a signaling pathway after adrenergic signaling. So they make a lot of sense in the activation of this canonical pathway of thermogenesis. And this is what I show you here. So the ADCY3 is placed here, and PKA again is this catalytic subunit that controls phospho-CREB activation, and also this controls GSP1 expression. We think, and actually this has been also previously shown that these genes are involved in obesity or metabolic regulation, and also more recently a paper from Philippe Frogel in London. They showed the ADCY3 monogenic mutation score cause severe obesity. So what we think it happens in the context of YY1 is that YY1 then, when it is dephosphorylated, serves as an enhancing by a positive feedback loop these signaling pathways by leading to higher amounts of ADCY3 and PKA2B by, again, as I said, a positive feedback loop of adrenergic signaling. Then if I have a bit more time, then obviously this is what we think it happens, that the YY1 phosphorylation controls anabolism and catabolism pathways, and somehow we are delineating its mechanism of action. But also we need to understand how YY1 phosphorylation is controlled at the signaling level. And I don't have, again, too much time to explain this, and I will have to show you just quickly the models, but not the whole data sets. And we showed that actually YY1 is phosphorylated by CK2, and upon cell GMP signaling, there is a dephosphorylation, and this is mediated by classical phosphatase PP1. On the other hand, this is not the whole story, because we wanted to be more precise and to understand a bit more how PP1 may control the dephosphorylation. PP1 phosphatases are rather promiscuous. There are not many phosphatases compared to the amount of kinases that are in the genome. So their specificity is less understood, and it's mostly thought that actually regulatory subunits of PP1s or phosphatases actually control the activity of the phosphatases, or maybe either positive or a negative way. And so that's what we are asking, what are these other potential regulators? And we focus on trying to understand a bit more the phosphatase aspect, because we think it's what triggers catabolism. And then anyway, CK2 also have explored it, but I'll show you for now a bit more this aspect of PP1. So for that, we actually used public databases to look for phosphatases or regulatory subunit of phosphatases that were upregulated upon cold exposure and in mice. And actually one stood out, and you see here by different times of cold exposure, we see PP1R3B highly upregulated upon these time points. And we looked a bit more in detail at this protein, and it seems to be highly expressed only in liver and brown adipose tissue brown, more specifically. I don't have the time to, and we also confirm this data. You can see here how in brown adipose tissue is highly expressed, but not in other white adipose tissues, and also in the liver is highly expressed. It's highly expressed in mesenteric white adipose tissue, but we don't know yet the function there. And we have done also other experiments, but we were actually very lucky and surprised. As a first approach, we wanted to see, okay, what does PP1R3B function? How does it control PP1, and maybe its potential targets? And by just looking at the biogrid databases, we saw that actually among the few known targets of PP1R3B or T1 interactors, there are at least five, six known. YY1 was one of them, and we confirmed this interaction. Actually, I don't show you this here, but because I want to show you a bit more precise other data. But we think that we showed that YY1 interacts with PP1R3B, but then what we did is to knock down PP1R3B to understand the phosphorylation of YY1, which is the more interesting aspect. And we see here, and we also have more data in vivo now, and because the reviewers asked us, and was also anyways planning to do it, we see that when we knock down PP1R3B, we see a dephosphorylation of YY1, suggesting here that actually PP1R3B blocks the activity of PP1, which is a bit contradictory or counterintuitive what we were actually looking at the beginning. We're looking for phosphatases that actually dephosphorylate YY1, but here we found a negative regulator of a phosphatase in this case. And also, interestingly, we see when we knock down PPR3B, we also see by the protein level an increased expression of UCP1, and it's highly nicely correlated, and also by mRNA expression. And we also, when we overexpress, we can also overexpress PPR3B. There is no change in UCP1. Yes, and this maybe I skip it. So the model stands as we think that upon there's an interaction of YY1 to PPR3B, and this blocks. Sorry, this actually inhibits PP1, but upon cyclic AMP, there is a dissociation between PPR3B and YY1, and this leads to the activation then by PP1 to dephosphorylate. So this is actually a mechanism that happens in the liver. So for glycogen synthase, PPR3B also blocks the activity of glycogen synthase by inhibiting the dephosphorylation. So this is something that is not completely unusual. It happens in biology before in other contexts. So we think also this is a similar mechanism, but here in the context of YY1. Again, here by dephosphorylating YY1 by this mechanism, then we see increase of UCP1. We took it then to the next level. Do I have still time? I lost a bit track of the time, Francesco.
Well, it depends how many slides you have, but yeah, you have some time. If you want to.
No, I have 2 more slides.
Yeah, sure.
2 more slides. Okay.
Sure.
Good. So just also to finalize, obviously wanted to go a bit more in vivo, and what we did is to inactivate PP1R3B in mice by using antisense oligos in collaboration with Ionis, and we knocked down, as you can see here, by injecting subcutaneously, and we see a strong reduction of PP1R3B by RNA, but also by protein. And also we saw a slight increase in body temperature, even though it's a slight increase in body temperature, it can be biologically meaningful in the context of thermogenesis. But more interestingly, what we saw also is a reduction in body weight upon high-fat diet. You see here total body weight, how mice are not gaining weight upon high-fat diet when we knock down PPR3B, and also just total body weight gain is dramatically also changed. And you can see here a picture of the total whole body of the mice. More interestingly, also this was associated with a decreased fat mass and lean mass and different also. So no change in lean mass, sorry, but change in fat mass in different fat depots, which actually I missed now that. Yeah, so this is grams of fat, and this is percentage of fat. This is lean mass, no change, and this is just total depot of white adipose tissue versus body weight, and this is inguinal adipose tissue, and this is a visceral adipose tissue, epididymal white adipose tissue. There's always a decrease in fat mass, depending on how you look at that. And finally, also we saw an improved glucose tolerance by GTT. Also, again, these mice were high-fat fed, and we saw a strong reduction in glucose or insulin resistance, let's say. And there's also a decrease in serum triglycerides by knocking down PPR3B, and also some genes that were associated to fatty acid synthesis like ACLY or CCC1 or FSIN were suppressed and not giving rise to a higher fat accumulation. So I finish here with the model again that I more or less advanced at the beginning. So we think that this acute adrenergic signaling leads to actually a positive feedback loop controlled by YY1 in the unphosphorylated state, leading to a catabolic and thermogenic state. On the other hand, upon actually a chronic cold exposure, this is a bit more complicated because I didn't maybe show completely all the data, but upon chronic cold exposure, then we see that the PPR3B is maybe attenuated and attenuate the dephosphorylation by recruiting YY1 and preventing the activation of these genes. And this actually dissociates, and this is, I mean, maybe hypothesis, dissociates the loop that is formed by the dephosphorylation. And in this case, these genes cannot be activated. By this happens, I think, by chronic cold exposure, again, to attenuate exacerbation of catabolism. On the other hand, upon high-fat diet, also we see that there is an increased phosphorylation of YY1. This is mediated by CK2, although I didn't have time to show you the data. and this activates ribosome and translation genes. And just to finish, so I showed you overall that Y1 senses adrenergic signaling through this dephosphorylation and orchestrates transcriptionally this thermogenic and catabolic program. The phosphorylation promotes the opposite, an anabolic phenotype through constitutive binding to promoters of ribosomal and translation genes. And we identified the PP1R3B as a key regulator that is induced upon chronic cold exposure, but we think we can also modulate this in the other different contexts and maybe trigger it and knock it down to promote energy expenditure, because we also saw, because in mice, when we reduce PP1R3B, these are protected against diet-induced obesity and insulin resistance. So I finish here. These are the people who did all the work, I have to say, as strong and talented PhD students. Sianya, mostly Sianya was involved in that, and Tian a bit, so Sianya Dia and Tian also helped at some parts. Chiara Safidi, a master student, was also involved in this project, and Monica Frey, lab manager, and Jasmine was also a master student. And we have also some collaborators who helped us with the analysis of the high C and for this chromatin looping, Bartel Planck at the PFL, and others at Florida State University and Karolinska Institute and Ionis for the ASOS. Good, thanks a lot. I'm happy to take any questions. Yep.
Thank you, Francisco. It was very nice mechanistic overview and beautiful paper. So the last slide means that you believe in women. So your team is, which is great, Obviously. Yes. So fascinating talk. I have a quick question and then I leave space to the others. How selective is the modulation of PP1? How do you think this approach could work in humans? So if you modulate PP1, do you think that downstream, what is going to happen apart from YY1? Yeah, that's a good point. So, and that's why I think it may be interesting to approach this in a selective way. And obviously PP1 is highly expressed everywhere, it's very promiscuous, but we found PP1R3B as a specific modulator of PP1, and PP1R3B is only expressed in brown and liver, brown deposition liver. So, and ASOS are already actually used and potentially in the clinics, or at least in advanced clinical trials. And knocking down PP1R3B in patients maybe would be potentially a way to increase catabolism and to improve metabolic and energy homeostasis. So again, just because PP1R3B is selectively expressed in these tissues, we could modulate this in a precise way. And this is a way to indirectly activate the downstream effects of YY1, because again, also YY1 is difficult to target pharmacologically or at least at the chromatin level, because YY1 is ubiquitously expressed and it's not an attractive target. But then PP1R3B, yes, we could target it and would indirectly then mediate the effects of with that YY1 would do. Yeah. Yeah, but this is really fascinating, and also we could also link your investigations to cardiovascular disease via the adrenergic stimulation. So there would be a lot to investigate in heart failure, for example. So and blood pressure regulation. So indeed, there is a question related to this from Dr. Gauscher. I say, very nice talk. In mice tissue is YY1 circadian. So does it change with time of the day and with the metabolic state? And is YY correlated with body temperature? Yeah, good questions. There's no studies showing this at least so far, and we haven't initiated any of those either. I have to say that I suspect it's not maybe that regulated, that at least its expression in circadian way, but maybe its regulators are, because Y1 is rather stable. And as you also saw by the chipstick, there's not many changes in Y1 binding, although some selective changes, yes, but we think it's maybe more the regulators and the post-translational modifications that then control the actions, but maybe not Y1 itself. So I suspect not, but it's just a pure speculation. And the other question was the body temperature, right? Well, the cold. Yeah. So it's either or same. It's not really also regulated by cold exposure or its levels. But this we have checked many times. So Y1 levels remain constant. But what we saw is actually a post-translational modification change upon cold exposure. Excellent. There is one more question concerning the phenotype of your mice in the ASO. It's the phenotype due to reduce. Do you think could be the phenotype you observe due to reduced fatty acid synthesis in liver because plasma triglyceride was massively reduced?
Yeah, that's a good point also, and could be because also PPR3B is expressed in the liver. We haven't seen, however, changes in any metabolic genes so far in the liver when we knock down PPR3B in mice. So we see gene expression and no change in FSAN or other genes involved in triglycerides. So yeah, we think that the triglyceride decrease is due, hopefully, we have to still validate this, but hopefully to an increased catabolic activity of brown adipose tissue that clears free fatty acids.
Yeah, it would be also nice to look at the cardiac lipidome in these mice, because I think that it could affect also the lipotoxicity there.
Yeah.
So yeah.
Sorry, may I say something? So we also have a transgenic mouse overexpressing PPR3B. So we are not only doing this model of knockdown with ASOS, we thought it was a fast approach and quick approach and also somehow translatable. But also we have overexpression models and also knock-in models of Y1, say 120. So we have different models to study that, yes.
So there is quite a lot to so we have to stay tuned, I guess, on your mice. Okay, so if there are no other questions, I think it's time to move on. So Francisco, thank you so much for this mechanistic study. It's really brilliant. And
thanks a lot for the invitation.
Joining.
Yes, let me just quickly stop this sharing the screen sorry, my screen.
So I would like to introduce the last but not least speaker, who is Emma Louise Robinson. Emma is an American Heart Association postdoctoral fellow. She works in the lab of Professor McKinsey in the Anschutz Medical Campus, the University of Colorado, United States. She's among the founders of the EU Cardio RNA Network, and today she will actually present part of a program in this direction. And her talk is entitled Leveraging Non-Coding RNA to Fight Cardiovascular Disease, the EU Cardio RNA Network. So we are really eager to hear also from the non-coding side, because this is still a lot to know about epigenetic regulation. And thank you, Emma, for being here. And please, stage is yours.
Thank you very much. Can you hear me okay? Can you hear me?
Yes.
Okay, great. Thank you. Yes, thank you to the organisers and the other panelists for this great first event. And yes, today I'm going to talk about a little bit about the EU Cardio RNA Network. And if I may actually start my talk, if I can move my slides. There we go. And if I may actually start my talk with a little bit of data from my project that is still in its relative infancy compared to some of the amazing projects we've seen today, but it was actually partly funded by the EU Cardio RNA Network, as well as using some of the active motif kits. So I thought I'd like to share it. And then I'll talk about the Cardio RNA Network. So just introducing the disease that I was studying in my previous postdoc, called heart failure with preserved ejection fraction, which many of you are probably familiar with, or HFpEF. And HFpEF is a complex, systemic, highly heterogeneous disease. It's associated with comorbidities such as obesity, type 2 diabetes, and hypertension, as well as kidney disease. And it's thought that these comorbidities contribute to both organ as well as systemic inflammation, which then causes, which then reacts on the heart, maybe through the systemic inflammation, causing changes in the heart, including cardiomyocyte hypertrophy, somewhat through titin stiffening, so changes in titin isoform expression, as well as post-translational modifications, and metabolic changes, including lipotoxicity. It's also implicated with fibrosis, so increased collagen and extracellular matrix expression, as well as composition, so causing stiffening, endothelial cell dysfunction and capillary regression, as well as there actually then being inflammation in the heart itself, contributing to dysfunction. And the result is a heart that is stiffer, so it cannot fill up as effectively during diastole, which has a result of decreased cardiac output, similarly to HFrEF, but it's an inability to fill up rather than inability to pump. And unlike the sort of classic HFrEF, the toolbox we have to treat and as well as diagnose HFpEF is relatively empty compared to that of HFrEF. There's been a number of trials so far looking at classic heart failure drugs such as ACE inhibitors and beta blockers that have been poorly effective to treat chronic HFpEF patients. And another thing to note, importantly, is that the majority of HFpEF patients seen in the clinic are actually females. So quite consistently throughout the globe, about 2/3 of HFpEF patients are female and older females as well. Of course, we have to note during the slide that in the last few weeks, there has been the first drug approved by the FDA to treat HFpEF, the SGL2 inhibitor brought about through the series of EMPEROR trials. So this is promising and actually the first drug that's been approved specifically for HFpEF. So I mentioned that one paradigm for HFpEF causation is systemic inflammation. And it's been shown by numerous groups that there's increased white blood cells in patients with HFpEF. And this group, our collaborators actually at the University of Dublin, showed that there was increased numbers of CD14 positive monocytes in the circulation. And even with people with the same comorbidities. So we've got asymptomatic hypertensive people, asymptomatic people who have some diastolic dysfunction but don't yet have full-on heart failure, and then those with HFpEF, and they could see that there was a gradual increase in circulating monocytes even between the asymptomatic patients. And so we're now looking to white blood cells not only as a causation and potential therapeutic target for HFpEF, but also as a source of biomarkers. And this is actually a project I worked on, which was funded by the Dutch Heart Foundation, called the CVON Early HFpEF Consortium during my previous postdoc. And one of our tasks in the basic science work package was to look at CD14 positive monocytes from HFpEF patients as well as appropriate controls, and perform transcriptomics and proteomics to look for, as I said, both biomarkers as well as potential therapeutic targets for HFpEF. So this was RNA sequencing, which is what I'm going to talk about today, from the CD14 positive monocytes in both male and female HFpEF patients compared with the at-risk controls. And the idea of this is that we're not introducing the comorbidities necessarily as a confounding factor, but actually the heart failure signs and symptoms. And this is the PCA plot focusing on the coding RNAs, the protein coding RNAs from the RNA-seq. And you can see here that there's some fairly stark clustering between both males and females, HFpEF, as well as males and females at risk, and between the disease types, between disease and at risk. However, when we then looked at the long non-coding RNAs, we performed total RNA sequencing. You can see the clustering both between the sexes and between at-risk and HFpEF is even more stark. And I've not quite ever seen clustering so stark in sort of four corners of the grid there. So we thought maybe the long non-coding RNAs may actually underlie the both sex specificity as well as the disease phenotype, at least with regards to inflammation, a little more than the protein coding RNAs. And we started to examine some of these differentially expressed long non-coding RNAs. So the one that I decided to focus on, and I'm going to talk a bit about today, is called Titin Antisense 1. And it's actually annotated in the human genome, but it's not characterized. We don't know what its function is. And it was upregulated specifically in the female HFpEF patients, and not so much the male, but there is a slight increase. And you can see here, this is the RNA sequencing alignment. So in white blood cells, as you would expect, titin itself is not expressed, but titin antisense is. This also is promising in that titin antisense is not just expressed as a byproduct of titin expression, it's actually independently regulated and present even when titin isn't. I wanted to validate, first of all, these findings. So this was the Dublin samples that I mentioned, on which we performed the RNA sequencing. And then we collaborated with Hester de Rauter and Saskia Jager in the University of Utrecht in the Netherlands. And they have it's a slightly different cohort. So this is called the Helpful Study, and it's actually less severe patients. This is more of a general population study. So the HFpEF patients weren't as severe as they were in the previous study, but you can still see here by qPCR that there's an increase in titin antisense expression in the HFpEF compared to the no LVDD individuals. And when we separated this by sex, this difference was predominantly seen in the females rather than the males. So thinking about whether this is obviously titin antisense is opposite titin in the genome, is this also expressed in the heart, where we know titin is very highly expressed? And I was very fortunate to collaborate with Nazar Hamdani, who's at Royal University Bochum in Germany, and Arantxa Miko, who is in Navarra in Spain. And they have access to endomyocardial biopsies from patients with HFpEF and HFrEF, or in the case of Germany, and Arantxa had samples just from HFpEF patients. So I examined titin antisense expression in these, and it was actually even higher than it was in the white blood cells. And you can see here again the increase in titin antisense 1 expression is seen primarily in the HFpEF females as opposed to the males, and again, more so than the HFrEFs. This is also a nice comparison here that we didn't see induction in HFrEF. Thinking about whether this could be, titin antisense could be a biomarker in females for HFpEF. So we've looked in the white blood cells, and that, as I said, it can be a source of biomarkers themselves. However, an easier, more accessible source of biomarkers is, of course, plasma, where a lot of people look for biomarkers. So I wanted to see if titin antisense was also present in plasma, and it was. And these are actually some of the same individuals for which we had the cardiac data. And again, the titin antisense was elevated in the plasma of female HFpEF individuals rather than the males. Just to say that, as with a lot of RNAs in plasma, the sensitivity for detecting titin antisense was rather low. We did play about with things like digital PCR to try and get more sensitive and quantitative data. But we're probably in the future going to look more to the white blood cells as a source of the titin antisense rather than the plasma. And I'm also collaborating, as I say, we're collaborating with the Helpful Study in Utrecht, as well as collaborating with Roger Fu and Mark Richards on the SHOP study, which is based in Singapore. So again, having very different populations of HFpEF patients between continents even. And we're looking at titin antisense expression in plasma and white blood cells in these big cohort studies, and overlaying it with the clinical data, which is taking some time, as you can imagine. So back to titin antisense in the heart and what it might be doing in the heart in HFpEF. So as I explained before, in the white blood cells, titin isn't expressed, but titin antisense is. And you can see here in the heart, as you would expect, lots of titin expression and titin antisense expression as well, but not at the same levels. So again, not indicating just a byproduct of titin expression. So I wanted to then see if titin antisense was also upregulated in the hearts of models, preclinical models of diastolic dysfunction. Obviously, if we want to start studying its function in the heart in HFpEF, we want to see if it's also present in our models that we use. So I looked both in the ZSF1 rat, which is a cross between the ZDF rat, which has a leptin receptor deficiency, and a spontaneous hypertensive rat. And this cross causes a rat which has hypertension, type 2 diabetes, hyperlipidemia, and develops diastolic dysfunction within sort of 12 to 16 weeks. And also very lucky to collaborate with Dirk Dunker's group in Rotterdam, who developed this wonderful pig model of diastolic dysfunction. And this pig has diabetes mellitus, which is induced by I can never say this word, streptozotocin, that ablates the islet cells, and chronic kidney dysfunction, hypertension caused by a renal artery embolism, and high-fat diet and salt to induce hyperlipidemia and obesity. This model actually only works in female pigs, interestingly. So that's one caveat here, is that I only actually have female models. But titin antisense is upregulated in the hearts of both these models. And in the pig, I actually had to go and find it annotated myself, because it's not annotated in the pig genome. So it's good to know that for future work, it's actually expressed also in these models. So one obvious question when you're thinking about antisense RNAs is whether they act by complementary base pairing to inhibit the expression of the gene of which they're opposite, in this case, titin. So I did some correlation, which I haven't shown here, of lots of RNA-seq data, both our own as well as publicly available RNA-seq data sets. And it seemed as though there was actually more of a positive correlation with titin than a negative correlation between titin and titin antisense expression. So it didn't seem to be just a basic inhibition of titin RNA. So I then went to look at whether it affects titin splicing. So titin, for those of you who don't know, is, I think, the biggest or second biggest protein, and has two main isoforms, the N2BA and N2B. N2BA is the longer, more compliant form of titin, and N2B is the smaller, stiffer form of titin. And it's been shown that in HFpEF, as well as other cardiac conditions, there's an increased expression of the shorter, stiffer version of titin, which contributes to a stiffening of the ventricle. And so I correlated both in human biopsies as well as in the pig heart. And there was actually a correlation between titin antisense expression and the amount or relative amount of the stiffer, shorter form of titin. And this was mostly stark in the females, a little bit less of a strong pattern in males, interestingly. So the main, at least I think the main or one of two known regulators of titin splicing is called RBM20, which is a very well characterized protein, and there's also mutations in which cause dilated cardiomyopathy and other cardiac dysfunction. And so I wanted to see if titin antisense associates with RBM20. And you can see here, this is actually in the human biopsies again from NASA. In the control, as well as the HFrEF and HFpEF biopsies, there's very strong association between RBM20 and titin antisense 1. This wasn't particularly different between HFpEF and HFrEF, but what's important is, I think, is that titin antisense is certainly binding RBM20. However, again, this could be just a sort of, an association, direct association, coincidence, because RBM20, if it's sitting on the titin, ready to splice it, it kind of makes sense that titin antisense would be in the same vicinity. And so maybe this is just direct and doesn't actually mean anything functionally. So I then went on to knock down titin antisense 1 in adult cardiomyocytes from mouse. And you can see here, that took a lot of attempts to get this knockdown to be efficient, but I managed to get an 80% knockdown of titin antisense 1 using a couple of different GAPMers, and then stimulated the adult cardiomyocytes with endothelin 1, which is a pro-hypertrophic agonist. And this is looking at some classic pro-hypertrophic genes. So MPPA or ANF is increased with the control with endothelin 1, but subdued when we have the titin antisense knockdown. Similarly for NPPB, which is also BNP, MYH7, not too much of a change there. And then skeletal muscle actin, which is also a signal indicator of pathological hypertrophy, was attenuated when we have titin antisense knockdown as opposed to the control scrambled GAPMer. Importantly, also titin isoform switching was seen. So when we just stimulate with endothelin 1, there wasn't a great change, but when we knocked down titin antisense 1, there was actually an increase of a longer, more compliant form of titin relative to the shorter, stiffer form. So maybe this is actually functionally affecting titin splicing in the heart, in cardiomyocytes. And just to say again, this is, as you can see, it's pretty early days that we're gathering this functional data as to what titin antisense is doing in the heart. This is one potential model. So when we have a healthy heart or no stimulus, titin antisense expression is lower, titin's being expressed, and we have more of the longer form compared to the shorter form. And we've got the healthy heart, which is not so stiff and elastic and can fill up effectively during diastole. However, when we have a stimuli such as inflammation, diabetes, obesity, aging, potentially female sex, which I'll talk about a bit more in a minute, we have increased titin antisense expression, which then associates with RBM20, affecting titin splicing and increasing the relative abundance of the short N2B form of the stiff titin compared to the longer, more compliant form, resulting in stiffness of cardiomyocytes in the ventricle and diastolic dysfunction. So I'm interested also in how or why this seems to be upregulated, particularly in female hearts and white blood cells and plasma as opposed to male. And particularly interesting is that I think all of these samples, at least from the humans, the HFpEF samples as well as HFrEF, were from postmenopausal women. So it's not an obvious direct effect that estrogen is upregulating titin antisense for some reason. So one hypothesis I had was that whilst estrogen may not still be present or particularly highly present in postmenopausal women, maybe there's an effect, an epigenetic memory of having had exposure to estrogen. So I designed a rather rudimentary experiment, started off with HeLa cells, we're now doing it in cardiomyocytes, where we treated the cells with either estrogen or testosterone, and then removed the hormones, and then waited another 72 hours before harvesting. And then we looked at different epigenetic marks along the promoter region of titin antisense 1 to see if there was any difference long term, even after removal of the hormones in the cells. And what we saw, and this is where I used the active motif MEDIP kits, which I'm a big fan of, we saw that actually, when we treat with sex hormones, in some of the areas, we saw a decrease in DNA methylation. So DNA methylation in the promoter is generally associated with gene repression. However, in the central portion of the promoter, there was an increase in DNA methylation after testosterone treatment, after we've even removed the hormone 72 hours later. So this was the main sex or hormone difference that we saw here. So in fact, in this case, it indicates it may be actually presence of testosterone rather than absence of estrogen that may cause differential methylation of the promoter of targeted antisense 1. And we did actually look at several different modifications, and this was DNA methylation was the one that showed this difference. As you can see, it's just at the moment preliminary, and we're following up on this also in cardiomyocytes with a stimulus like isoproterenol or endothelin-1. Okay, so future perspectives for this project, which is being carried on by a couple of PhD students who are still in the lab in Maastricht, is to then look at different preclinical models of HFpEF and HFrEF, and we know it's expressed in the heart in our preclinical models, and knock down titin antisense 1 with GAPMers or overexpress it, and we have a AAV29, which is a cardiac-specific overexpression vector, and to see whether this is detrimental or beneficial in vivo. Okay, have I got time left? I know we're kind of tight on time. Okay, I'll just tell you a little bit now about the EU CardioRNA network, which is called Catalyzing Transcriptomics Research in Cardiovascular Disease. And as many of you who are involved in European projects may know, when there's often involved, lots of acronyms are often involved. So I'm going to go through a few of the acronyms first of all. So EU CardioRNA network is called a COST action. And COST stands for Cooperation in Science and Technology, and it's a scheme funded by the European Commission. And this has actually been going on for a number of years now. I think they were founded about 30 or 40 years ago, I believe. But I think there's certainly more and more funding going into this scheme, because the nature of science is changing and work is becoming more interdisciplinary, collaborative, data intensive. And so collaboration is, of course, an excellent way to solve some of the unmet needs in science and technology and to solve complex problems. And the COST action funds not so much individual research projects, but it funds networks. And it does have some small grants other, not just academia. As I said, it's funded by the European Commission, so it was funded by Horizon 2020, which is now Horizon Europe, and the headquarters are in Brussels in Belgium, which I've been very fortunate to visit and had some training there myself. So CardioRNA was a project that was started in 2018, and they're generally 4-year projects, so we'll finish in 2022 next year. And the scope is to increase multidisciplinary work to accelerate the understanding of transcriptomics in cardiovascular disease, and especially to further the translation of experimental data into usable applications to improve personalized medicine. And it's predominantly a pan-European network. Of course, it's funded by the European Commission, but we do also have international partner countries, including the USA, of course, where I am now. And our team consists of researchers, clinicians, industrial partners, also people involved in policy making. So it was approved in 2018. We had a kickoff meeting in Brussels in October 2018. It's due to finish in October 2022. When the project started, we had 24 countries involved that signed the MOU, which stands for Memorandum of Understanding, so basically the contract to which you sign to go into the project. And we now have more than 37 countries from all over the globe involved. So our network has really grown, and actually especially in the last year. And this is our website down here. So to introduce the team, the leaders of this network, and I'm going to try not to get emotional about this. So some of these, the leaders here have really been incredible mentors the last few years, and I'll be very sad when this project finishes. But the chair of our network is Yvan Deveau, who is based at the Luxembourg Institute of Health. Costanzo Emanuele is our vice chair based at Imperial College London. Ines I can't say her name, Potonjak, who is based in Zagreb in Croatia, is our grant holder and scientific representative. Dimitri Base is our SDSM chair, which stands for short-term scientific missions, which I'll mention in a bit. I'm the science communications manager, and Inga Dardesheid is our science officer, who is actually based at COST. So she works for COST itself. And we have 4 work packages. Work package 1, which is regulatory function of the transcriptome. We have work group 2, which is best practices and experimental standards. Work group 3, which is development of cohort inventory database. And we also have a dissemination work package, which I head up. So as I just described, these are the work packages and setup, and we all work together. Obviously, we need to disseminate the work of all the 3 working groups through working group 4, and we aim to disseminate and share our work with peers, with specialists, with clinicians, and policy and medical policy makers, as well as industry and funders. So just to go through a few of our activities, we have meetings, whole action meetings, 1 or 2 per year. Obviously, we haven't had one since Maastricht at the beginning of last year. And actually the core group here are now in Athens meeting, and I'm very jealous. They're having their first in-person core group meeting as we speak in Athens. The network funds short-term scientific missions. So these are exchange visits for researchers to go to another lab that is involved in the COST network for anything from 3 weeks to 3 months to start a collaborative project. We also have inclusiveness target country conference grants. So within the COST, there's a number of countries which have been identified as maybe not having such research, being not so research intensive, not so much money from the government being able to go into research. And so we support these countries and give their researchers more of a chance to visit conferences when they might not be able to otherwise. We also organise training schools, and again, there's one going on right now actually in Norway, teaching the younger members of the COST Action about gene expression analysis. And we've had so far 3 big in-person meetings, one in Lisbon, one in Istanbul, and then one in the Netherlands at the beginning of last year. So just an example of the kind of things we do together, or we have, there's been lots of spin-off projects. Importantly, we filed an application for another EU project based on COVID called Caverna, which was funded. So this is more than a £4 million initiative, euro, but very confused, initiative that was successfully funded. There's also been a number of other European projects, including some Marie Curie applications, which have actually stemmed from one of these STM awards. So Mila went to Luxembourg, spent a few weeks there, then they wrote a Marie Curie application, and she's now actually relocated from Bosnia to Luxembourg. We initiate special issues in a number of journals. We've had a lot of special issues so far, at least 10. We're also involved in writing position papers on the role of RNA, non-coding RNAs functionally, as well as biomarkers in complex diseases. And just to talk a bit about how we managed during COVID, which I thought was really, it was a real support network during COVID. And as I said, if we ever disregarded the value of communication and unity, then I think we haven't, hopefully, in the last year or so. So we weren't able to have in-person meetings. We were locked down. Some of our members were, in countries which really didn't deal very well with COVID and were isolated, living alone. And so we actually were very productive during this period of time. As I said, we wrote a big European project. We had a number of papers published during the lockdown, both on COVID and otherwise. We also had 2 successful online meetings, which again, we had representatives from nearly all of our members, more than 30 countries, more than 100 participants in each one of them. We also reached out, sort of having a bit of fun, had a WhatsApp group during lockdown so we could share how things were going and try to keep each other's spirits up by sending photographs of our pets or bees or memes and things. So I think we really came together to try and keep up morale during this horrible time. Yes, so please connect and ask any questions and get in contact about the network if you'd like to join. We're on lots of social media. We have a LinkedIn group, Twitter. We have a project on ResearchGate where we collect all our papers. We also have a YouTube video if you want to find out more about what we do, and the website. And this is Yvan Deveau, our chair's contact details and mine. So please get in touch if you're interested. This is the thank you for the the work, the data, the total antisense data that I showed earlier, which was, as I said, performed in the Netherlands at my previous postdoc, and all the funding bodies and collaborators. And just as a final slide, I'm now in Colorado. I moved here actually almost exactly a year ago today, and I'm in the lab of Tim McKinsey. This is our view literally from our kitchen work, so across the CU Anschutz Medical Campus with the Rocky Mountains in the background. And this is a view from the Red Rocks Amphitheatre. And this is our group. This is Tim, my PI, and our wonderful lab members. And just a quick shout out that we are recruiting for postdocs in Colorado. It's a lot of fun, and we work on gene regulatory mechanisms and cardiovascular disease also. So if you're interested, again, please give me a shout. Thank you.
Great. Thank you, Emma, for this fascinating journey, starting from your investigation of PEF. It's really interesting. And I've seen you have established a huge network over the years and during your postdoc, so this is really impressive. And the cardio RNA network is also attractive. So I think it's, please, if you have questions and if you want to connect with Emma, do not hesitate, as she has, I think, a lot of things to say. So maybe perhaps we have space for one or two questions. One question coming from the audience is, what is the impact on gene expression, fibrotic hypertrophic markers, if you transfect a cell line with titin AS1?
If we overexpress it? Yeah, we don't know yet. We don't know. So we've had this AAV made to look in vivo, but we haven't looked in vitro yet at the effect of overexpressing it. No.
And so it's a great study. FPEF is also associated with plasma volume expansion. Does that mean that multiparity increase the risk of FPEF in postmenopausal women?
What's that? Sorry.
Does that FPEF is associated with plasma volume expansion? Does that mean that multiparity increase the risk of FPEF in postmenopausal women?
I'm not sorry. I'm really sorry. I'm not sure I quite understand the question. But
yeah, if they say if these women have maybe several gestations, or if this is affecting the.
Okay, understood. Yes, that's not something I actually have information on, whether their patients have been through childbirth. But it's a very good point. Of course, it's also interesting that if they have, they probably went through a series of physiological reversible hypertrophy at the time, and that's actually not information we have from our patients. And the animals we used haven't been through childbirth; they've just been bred with the study. So yes, it's good. It's a good point. It's good information to have from the patients, but we often don't have it.
I also have a question. So you focus on the transcriptional regulation of titin. So how do you see transcriptional regulation versus post-translational regulation of this protein in FPEF?
Of titin?
Yeah.
Yeah, so we haven't yet looked at post-translational modifications of titin when we knock down titin antisense 1, for example, or maybe overexpress it. And we're looking to collaborate with Wolfgang Linker, for example, who can look at titin. I understand it's a very difficult protein to study. So we're looking to collaborate to look at post-translational modifications, and we may wait until we have the animal models finished for that. But yeah, it's a good point.
Great. I've seen you have the right collaborators for what you need to look at. Fantastic. So with this, I think it's time to have a concluding remarks of this meeting. I see there are no additional questions. So thank you very much, Emma, for taking the time, for waking up so early today. And so I will share my screen to have a concluding remarks of this. And so it was really a fantastic event. I'm extremely happy of the way it went and the number of people and the quality of the questions that we've got. We got excellent scientists from the other side. I also learned personally a lot from this, from the speakers, from the active motive and the opportunities they've offered today. I think there is definitely room for a series of meetings in this field. I'm sure that next year, maybe if we have, with a little bit of optimism, we can organize a face-to-face event. Why not? And since Sarant is from Greece, I would propose Santorini.
Why not? Why
we
not?
Have to be ambitious, you know, in terms of budget.
True.
No, but I mean,
to aim high.
Yeah, aim high always. So, and then, yeah, I would really look forward next fall, perhaps, that we get together again with, I'm sure there will be lots of new stuff. And I'm sure that this community that will grow quite fast in the next years. So I was personally very satisfied by this initial first event. I would like to thank again Active Motive for the support for organizing this, my team for helping me in the organization, Samuel and Sarah, and Shafiq for the data, and really thank you everybody for joining, and thanks again to the speakers for entertaining us with the beautiful science. So really hope to get in touch with you soon. And thank you, Sarantis. I don't know if you want to add something.
Thank you. And I would like to thank also Stefan. He did a great job behind the scenes and to run smoothly and he was more stressed than everybody because he was keeping the times, you know. And I would like to thank also Stefan and you guys for the participation. And I'm looking forward for the next event. So
thank you so much. So keep in touch.
Thank you. Bye.
Bye. Bye.